There is another approach to find the genes within the genomes of any organism. Previously, we have studied the statistical approach to find the genes. Now we will see the similarity-based approach to find the genes within the genomes of any organism. So in this approach, we make use of the previously sequenced genes and their proteins, and we match these sequences with the newly sequenced DNA sequences to find the similarity between the genes. So instead of using the statistical properties of the exon in the similarity based approach, we will consider the combinatorial puzzle to find the genes. And in this combinatorial puzzle, we will consider that there is a sequence of a gene, and we will consider that sequence and the protein that it codes. And we will find the substrings that will constitute the exons, and we also find the introns that will fit with the target sequence. We can make use of the brute force approach, and in the brute force approach, the alignment of the spliced sequences will be considered, and all the similarities at the local alignment level will be considered, and all the similarities will then be used to find the similarities between the substrings that were isolated and they were identified while using that brute force approach. So the genomic sequence that exhibit the sufficient similarity to the target proteins, that we call it the substring, is now known as the putative exon. And that putative exon that was identified by using the brute force technique and by identifying the substrings on the basis of local similarities, they might not contain the flanking regions that differentiate between the introns and the exons, and these flanking regions are AG and the GT. And we can extend or reduce the sequence of the putative exon until we end up with the flanking regions of AG and GT. This we may get the overlapping sequences of the putative exon, but our target is to find the non-overlapping regions of the exons. And because we consider only the non-overlapping regions, because we know that the exons, they are present independently and they do not overlap with each other. So we can model a putative exon with the weighted interval in the genomic sequence, which is described by three parameters: L, R, W. Here the L is the left-handed position, R is the right-handed position, and W is the weight of the putative exon, which we have identified by using the brute force approach. The weight W may reflect the local alignment score for the genomic interval against the target protein sequence, or the strength of the flanking acceptor and donor sites, or any combination of these and other measures. It reflects the likelihood that this interval is an exon. A chain is any set of non-overlapping weight intervals. The total weight of a chain is the sum of the weight of the intervals in the chain. A maximum chain is a chain that have a maximum total weight among all possible chains. Below we will see an algorithm that will have all intervals that are positive and W is greater than 0.